Today we'll be studying DNA. And DNA is made up of the same base pairs. It's adenine and thymine and guanine and cytosine, and those 4 base pairs are rearranged in millions of combinations, and that tells us what we are. Everything that's living, you, me, strawberries, plants beside me, all have DNA, and that's what codes us for what we are. Now, today I want to show you DNA. Well, you might say, Mr. Bigelow, DNA is so teeny tiny we need a microscope to see it, and that'd be normally right. But what we're going to do is we're actually going to take the strawberries here and we're going to break them down so you can actually see the DNA. And this is science that you can do at home. I'm at my kitchen table. These are all normal things that you can have. You don't have to have beakers. You can use plastic cups. It doesn't really make a difference. So what do we need to do this? Well, first, this is about 3 strawberries cut up here. We use strawberries because, one, they're easy to get. You can use fresh, you can use frozen. It doesn't make a difference for this experiment. And strawberries also are octoploids, which means they have 8 sets of their chromosomes. That's a whole lot of DNA. Humans, we have 2 sets in our cells. So we've got 4 times as much in the cell of a strawberry as we do in the cell of a human. That gives us a lot of DNA to work with. Other than that, we need about a half a cup of water. That's about 140 milliliters. We need some salt. This is 1 teaspoon of salt, which is about 5 milliliters of salt. We'll use 2 teaspoons of dish detergent, dish soap. And then we also need isopropyl alcohol. This has to be really cold. So this has been stored in the freezer. I don't know if you can tell, but it's actually frosting up now that it's out of the freezer. You need to be really cold for this experiment. You can't use room temperature alcohol. I'll need somewhere to be mixing things. And then we also need something to filter with. I've got a funnel. This is just a coffee filter that we'll put inside. You can use cheesecloth. If you've got a really fine sieve at home, you can use that. We just need something to be able to filter with. And then when we're all done, we'll need something to get that DNA out. You can use tweezers. You can use, I've got a glass stirring rod for chemistry. You can use a bamboo skewer or even a toothpick. It doesn't really make a difference. So the first thing we're going to do is we're going to take our strawberries. We're going to put them in a plastic bag. All right. And we're going to kind of squeeze the air out just so we don't have a balloon that we're working with. And then we want to mash those strawberries up. So we're just going to mash them. The mashing is mechanically breaking down the cell walls. Remember, our plant cells have a cell wall and a cell membrane. So we need to break that down as much as we can. So we just squeeze it. Give it to your little brother or sister. Let them smash it for a while. We want to break these strawberries down as much as we can. So as we're breaking down that cell wall and that nuclear membrane, it's releasing the DNA. Trying to break up all the clumps in here. You can see it's getting juicier and juicier as we work. All right. So now we need to make up our DNA extraction solution. And this is called a lysis buffer. To lyse something means to break it apart. So we're going to lyse the cells, we're going to break them apart to release the DNA. So this is where some of our other ingredients come from. So in my big beaker here, I'm going to mix up my other ingredients. We're going to add the water. And the salt. And the dish soap. The trick with this is we don't want a lot of bubbles from the dish soap, so just kind of stir it up. Stir it up without trying to make a lot of bubbles. Now, why the dish soap and why the salt? The salt helps make an environment that the DNA will clump together. Okay? It actually breaks up the histone that's holding the DNA pairs together and allows it to separate, which then we can, once we've broken it all apart, we can make it clump together. And the dish soap is because soap is good at breaking up fats. And if you think about our cell membranes, most of the membrane is a phospholipid bilayer, which means you've got fats in there. And so the soap will grab onto those fats and break them apart, pull them apart, separating them further from the DNA. All right, so this is our lysis solution. So we're going to add that to our strawberries. Squeeze out the air a little bit. We're going to mix it up further. Once again, we're not trying to make a lot of bubbles. So we want it to break up and release that DNA. Okay, so now in my clean beaker, I'm going to take my coffee filter and the funnel. Now if you're really handy or you have someone to help, you don't actually have to have the funnel. You could hold the coffee filter down in it and pour your strawberry slurry into it. Since I'm by myself, I figure I will use the funnel to try to help hold things in place. So what we want in the bottom, you can see it pouring through. We want all the liquid from the strawberry, but we don't want the pulp. We don't want the solids. And this takes a little bit. You can kind of help it along if you carefully squeeze it down. Try not to make a mess in your parents' kitchen. So you can see I'm just carefully squeezing it. We want to try to get all the DNA that we can, since we worked hard for it. All right. All right, now we have our strawberry slurry. We want to add rubbing alcohol to it. Remember, this is a cold rubbing alcohol. Now it works best if you can pour it down the side so you're not just like dumping it in, but slowly adding it to your strawberries. And what we'll start to see is a separation inside the beaker. Okay, now you can see with a little closer shot the separation of the two layers. That's the alcohol on the top, and the pink below is the strawberry with the lysis buffer. And then floating on top, that white kind of snotty looking stuff, that's the DNA. So this is strawberry DNA. So here is our DNA, and that DNA, you could store it as long as you keep it somewhere cool. You could put it in another container or a Ziploc bag, and you can store it for a long time in the refrigerator. So all those DNA strands, we broke them out of the cells and then added our alcohol, and they kind of floated up to the surface. They precipitated, they came out of the solution. I shouldn't have to say this, but just as a warning, do not eat the strawberry DNA. So in the beginning I talked about using strawberries. Challenge you, you can actually use all sorts of different fruits to do this, okay? So if you've got bananas at home, use the bananas. You got raspberries or blueberries, use those. You can use lots of different fruit. You just got to be able to mash it up and to be able to get to our slurry here. So have fun with this.